Welcome everyone on behalf of the organizing committee of the Mathematical Science Colloquium. It is my pleasure to welcome our speaker today, Professor Genevera Allen from the University of Columbia in New York. Genevera just recently joined Columbia after serving as assistant associate professor in electrical and computer engineering, computer science at Rice. She was the founding director of the Rice Center for Transforming Data to Knowledge. So Dr. Allen received a PhD in statistics in 2010 from Stanford with Rob Tiprani. She works in the field of statistical machine learning and is known for her methods and theory in unsupervised learning, interpretable machine learning, data integration, graphical model, and high-dimensional statistic. Unfortunately, the talk has to be online because her flight got canceled this morning, but well, let's still enjoy the presentation. Organizational things. So if you have any trouble, please write in the chat or use the Q&A to ask your questions. I'll be monitoring those. Feel free to ask questions during the presentation. And you should be also be able to unmute yourself or just raise your hand. So thanks again, Genevera, for accepting our invitation. The floor is yours.
Okay, great. So I'm excited to be here today, but I'm very sorry I'm not there in person. Unfortunately, my flight was canceled this morning, so I couldn't quite make it, but I'm very happy to present online. So please feel free to ask questions throughout. I'd love to answer your questions in real time. So today I am talking about inference for interpretable machine learning and talking about interpreting features. So let's start with a little motivation. We all know that AI is being widely used, and it's being used in many aspects to make critical decisions in our lives, from healthcare decisions to personal finance, criminal justice, technology, self-driving cars, and so forth. And a lot of people have been asking, can we trust AI and machine learning? And one of the #1 things that people recommend to help develop trust in AI systems is to make them interpretable and explainable so that humans can understand what's really going on under the hood. But what we're going to be asking today is, can we really trust those interpretations? So is it enough to just make an AI system explainable or interpretable? Don't we need to go one step further and make sure that we're really understanding and trusting those interpretations that come out? So as we know, a lot of these AI systems and modern systems are developed on deep neural networks, and these are essentially black boxes. So the goal of interpretable machine learning is to uncover what's going on inside that black box. And I define interpretable machine learning as techniques to find human understandable insights into the data itself, the machine learning model, so this could be the fitted machine learning model, or the machine learning model's outputs. So these could be, for example, predictions or decisions of the machine learning system. And there's actually, over the past decade, been a ton of work in interpretable machine learning. This is a very active area of research, mainly in computer science, although there are some statisticians and mathematicians working in this as well. And I wrote a recent review paper outlining kind of many of the taxonomies of interpretable machine learning and specifically focusing on statistical challenges that come up in interpretable machine learning. So again, this is a very widely active area of research here. But we're going to focus specifically today on what I would argue is one of the most popular machine learning interpretations in supervised learning, and that is feature importance. The question is, how do the input features affect the model's output? Or you can think of it, how do they affect the predictions or decisions that are being made by the model? And feature importance is very widespread. There's a lot of techniques for this. So we're going to exclusively focus on tabular data today. By tabular data, I mean data in the form of observations by features. We can kind of think of them as a data matrix, as coming from a data matrix. So this is a bit different than video or image data, or perhaps sequence data in large language models. Okay, so that's kind of feature importance in those areas is another can of worms and is not going to be the focus of today's talk. So we're specifically talking about tabular data. You can think of tabular data as like electronic health records, where we might have patients by certain clinical variables and other diagnostics as features, okay? Or this could be genomics or omics kind of data, where we might have cells by kind of genes or gene expression or transcripts, okay? So, as I mentioned, there has been a huge literature on developing metrics and measures of feature importance. And largely, these can be broken down into model-specific or model-agnostic feature importance methods. Model-specific methods are methods that can only be used for certain classes of models. For example, statisticians are familiar with this. In linear models or generalized linear models, we can specifically look at the coefficients or parameter estimates in those models and use those as a measure of the importance of that particular feature. We can use the mean decrease in impurity for trees or random forests. And in deep learning and neural networks, there's a huge variety of methods based on gradients or input times gradients. Deep lift and layer-wise relevance propagation are very popular methods. And those are only applicable to neural networks. But what we're going to focus on today are model agnostic measures of feature importance. And model agnostic means that these measures are applicable to any predictive machine learning model, so any supervised learning model. And the advantage of model agnostic measures is they're comparable across different model classes. You can directly compare a feature importance in a random forest to something in a neural network to something in a linear model. And so that actually is a really nice property when we're talking about this. And there's three main classes of model agnostic feature importance method. They're permutation importance, and the idea here is that you shuffle or permute the values of feature J and understand how that affects predictions. There's Shapley values, and this assesses the predictive value of adding feature J to all possible coalitions or all possible subsets of features. And our focus today is on occlusion, which I would argue is the easiest of all, or the simplest to understand of all model agnostic methods. The idea here is incredibly simple. Remove feature J and understand how that affects predictions. So if we didn't have access to feature J when we were building our models, how would that change my predictions? And if it changes your predictions a lot, feature J is probably actually really important. Okay, so that's the idea of occlusion feature importance. Okay, so a very simple notion. We take away feature J, and how does that change my predictions? Okay. So there's all these great measures that have been developed of feature importance. Again, a huge literature on this, thousands of papers, especially in the computer science community, and a handful in the stat community. But the question here is, can we really trust these feature importance measures? Are they reliable estimates? Are they really estimating what we want, this quantity that we want? And specifically, we wanted to understand, are they reliable? And to motivate this, we actually did a very large-scale empirical reliability study where we specifically tested the reliability of feature importance through repeated random data perturbations of over 30 commonly used benchmarks in machine learning. We used 10 different types of data perturbation methods. You can just think of repeated sampling of a training and test set as one simple random data perturbation. We used over 50 different popular feature importance methods, and we used a lot of methods to measure the consistency or stability of the interpretations, several different metrics. So you can kind of see the study design here. We take many different random training and test set splits, and we apply our feature importance importance measures, and then we test how consistent are the features, important features across splits, and how consistent are they between different splits, and are they related to prediction accuracy. So just briefly, here's a preview of a tiny subset of the results. And what we're showing here is different benchmark data sets in machine learning. And on the x-axis are different interpretability methods. So these are different feature importance metrics. And it's showing the method and the interpretation approach. And the values you're seeing here are something called the rank biased overlap, or a measure basically of the consistency of feature importance rankings for these methods. So dark teal indicates very high consistency, and very light teal indicates very low consistency. And these are metrics between 0 and 1, 1 being perfectly consistent and 0 being perfectly imperfect. And we're showing also the prediction accuracy of these methods. And what we notice here, let's look in this kind of red boxed author data set, that the prediction accuracy across all these methods is really, really good. It's close to 1. It's like all these methods are really accurate. But look at these kind of interpretability, feature importance consistency rankings. They're all over the place. Some are a bit consistent, but some are really inconsistent. And this actually happens to be permutation and SHAP, some of these Shapley values, some of these model agnostic methods. So we're really seeing that while certain methods can be very accurate in predictions, their interpretations, meaning their feature importance measures, are kind of extremely variable. They're all over the place. And just to understand, are there any specific feature importance metrics that are better than others? This is a bump plot where we're showing the rank of the feature importance metrics across all these data sets. And it really kind of just looks like a pile of spaghetti or noodles or something. And this is really just saying, hey, there's not really any one feature importance metric that is super kind of reliable. Basically, there's a lot of inconsistency between these estimates of feature importance. And we might look between methods and say, okay, well, random forest and decision trees and XGBoost are all pretty kind of similar methods. They're all tree-based on methods. Maybe those methods should actually be yielding similar interpretations. So the idea is, should similar methods be yielding similar interpretations across methods? And the answer is no. So here this is presented in purple, and the consistency of the predictions is actually quite high. So the predictions are very consistent across methods, but the interpretations are all over the place. They're not consistent at all. And so kind of our conclusion, this is just a preview of this kind of large-scale study that we've been working on in my group. The kind of conclusion here is interpret these feature importances with caution, right? Like they're not as reliable as one would hope they are. And so this really motivates the need for uncertainty quantification. If our feature importance metrics are highly variable, and there's a great deal of uncertainty in them, we really need to quantify the uncertainty. And since I'm a statistician, we're going to do this via statistical inference. And the question is, can we do statistical inference for feature importance in machine learning methods? So the objective of my talk today is to provide confidence intervals for feature interpretations of any machine learning model. So these are model agnostic, can work with any supervised learning model, regression, or classification task, and without making any additional modeling or data assumptions. So this needs to be completely distribution free. We make no assumptions about the generating mechanism of the data or the model itself. And we're specifically going to talk about feature importance. So this is interpreting each single individual feature. And we're also going to talk about higher order feature interactions or patterns of features and their importance. And again, our focus is going to be occlusion-based importance for tabular data. And we're going to see that there are some benefits of using this occlusion-based formulation. Okay? So let's jump right into, and if there are questions, please do feel free to ask throughout the talk, okay? So let's jump right into inference for feature importances. So the question is, can we develop a distribution-free and model-agnostic inference method for feature importance? What we'd really like is we'd really like to get a metric that says how important each feature is attached with a valid confidence interval that quantifies the uncertainty in that feature importance metric. So related lines of research in the recent statistical machine learning fields are conformal inference for predictions, and these yield valid distribution-free confidence intervals for predictions. They're completely model agnostic, completely distribution-free, both of which we want. But so far, the work for this is really only focused on predictions, not on machine learning interpretations or feature importance. Another related line of work, of course, is post-selection inference. And in post-selection inference, part of the challenge with machine learning models is they're fit to the data. They're searching over many possible models. They're really model families, and they're selecting models based on the data via hyperparameter tuning. And this creates challenges, of course, for doing inference because we're not specifying our hypotheses necessarily in advance. And so there's been a great line of work on post-selection inference, and this is doing valid inference after model selection, sometimes conditional on model selection. However, these lines of approaches are really only model specific. They work only for specific machine learning methods, and you can't really apply them generally to things like neural networks or random forest or off the shelf any of these metrics. So there's another kind of lines of work, and we define kind of inference for feature importance in two categories: machine learning feature importance and population feature importance. So in machine learning feature importance, we want to know how feature J affects F hat, our fitted learned machine learning model. So this was trained on a data set. It's a learned model, and we want to know how feature J affects our predicted learned model. So this is how does my specific trained model use feature J to make predictions? And so this is a property of my specific ML model and my specific training set data interactions, okay? Now, on the other hand, there's population feature importance, and that asks a slightly different question. And that's, how does feature J affect F star, where we are going to define F star as the population model or the data kind of generating mechanism. And so this is really a property of the data itself and not the specific ML model that you trained on a specific data set, right? We just hope that we train an ML model that so that our F hat is close enough to F star so we can say something about F star. So this is slightly different. And population feature importance has been super well studied. This is all of classical statistics. All of classical statistics makes an assumption about the data generating mechanism, essentially F star, and does inference with respect to F star. And in the context of machine learning and specifically occlusion, there's been a very interesting recent line of work, and here are several papers that I can recommend here on actually how to do occlusion-based inference for the population feature importance that is applicable to things like neural networks and random forest. However, all of these models require very strong assumptions. You either need to make an assumption on the data generating mechanism, so some type of assumption on F star, or you need to make some very strong assumptions on F hat to ensure that F hat is close enough to F star. And those assumptions on F hat don't necessarily hold for things like neural networks or random forest or other really big machine learning models. So there's some caveats to these approaches, and I would argue that it's not really getting directly at kind of the machine learning model itself, F hat, and how to interpret that machine learning fitted model. It's actually doing inference on F star. Instead, the ML feature importance, there's really only been one approach that's been proposed so far, and this is called LOCO, or leave one covariate out. And this is going to be the focus and what we're going to build upon our inference procedures today. So LOCO is leave one covariate out inference. And this was proposed by Jing Lei and several others at Carnegie Mellon University. And the idea is to do inference on feature occlusion scores using data splitting and some tricks from conformal inference, this kind of predictive inference. And so specifically, the inference target is as follows. The delta J star is the expectation conditional on X and Y are our training set because we built the model on a specific training set. And it's the difference between the error between Y and F hat minus J and Y and F hat. So this is actually the original model that has access to all the features. And over here is the model that has access to all the features but feature J. So feature J has been removed or occluded or left out, and we're looking at the difference in the prediction error. So this error is just any prediction error metric. We're looking at the expectation of this difference over new values, actually, of X and Y. Okay, so that's what we're looking at here. And so you can interpret this delta J as the difference in expected prediction error with feature J and without feature J. And so if you stare at delta J long enough, it's the difference in errors, which we want errors to be lower, of course, prediction error. So delta J is going to be positive when feature J improves the predictions, and it's going to be negative if feature J actually made the predictions worse, okay? So the interpretation of this is how important is feature J when we're deploying our learned model F hat. When we have actually trained a specific model and we're deploying it in practice, how important is feature J? So a specific application of this might be a big bank who actually built an ML model for to predict kind of a personal finance decision, like applying for a credit card, whether you're going to be accepted or denied into a particular credit card. And they build this big machine learning model. And when they go to deploy this and actually use this in practice, this interpretation of delta J is how important is feature J to actually deploying this model in practice. So how important is something like credit history or credit score or income when deploying this particular model? So I would argue this is a really interesting inference target that was studied in this loco inference. However, the approach, oh, I see a question. I think let's go ahead and take the question. Yes.
Okay, thank you. Just to make sure that I understand, in the minus J model, you're not refitting the model. You're using the same model, but just removing the feature. You're not refitting the model, are you?
So for now, we're actually going to assume, so these original loco papers and our approach is actually going to be refit models. However, there are other ways of occluding feature J. So sometimes if the X's are centered, for example, you just set it to 0, right? So either one is okay, but we're specifically going to study the refitted version. So we're refitting F hat without J. So it's kind of like if F is the class of random forest models, it's like how well could a random forest do with feature J versus without feature J is the way to interpret this model. However, there are extensions of this to actually just occluding by kind of setting it to a null value instead of refitting the model. Great question. So, this group, so Jing Lei and others at Carnegie Mellon, their approach to do loco inference was something that we call loco split via data splitting. So they split their data into two equal halves, D1 and D2. You're going to fit the supervised model to D1, and you're going to use D2 to evaluate the prediction error and construct confidence intervals. So their inference target is a little bit different because it's conditional on half the data, okay? So you're using kind of half for fitting and half for inference or calibration. And you do really need both of those sets to be as big as possible, right? Because you need enough data to do valid inference and or correct inference, and you need enough data to also actually fit the predictive models. So there's some great advantages of this approach. This is truly model agnostic. However, the theory that was developed previously was only studied for regression. It's fairly assumption light and it's completely distribution free. So this is actually really nice advantages here. But there's some major disadvantages also. The data was split into half. And so this loses a lot of statistical power. If we can only use half of our data for training, we might not train a particularly powerful ML model that we want to deploy. Also, it's computationally pretty prohibitive. If we look at this following the previous question, if you actually have to refit a model p times, if we've got p number of features and we're refitting a model p times, that can be computationally pretty challenging, especially for large numbers of features. But I think the third point is actually most important. The inference depends on a random data split. So the first step is split your data randomly in half into kind of a training set and a calibration or inference set. And so your inference is conditional on a random split of your data, meaning if you split your data differently, you might actually get different inference. So can you imagine going to collaborators and giving them a confidence interval or p-value and saying, well, this is statistically significant, conditional on using this random seed in my program. And if I used a different random seed, I might get a different answer. I mean, to me, that's really philosophically unsatisfying, right? Like, because we actually want to use all the available data, and we want to use as much data as possible for training and for inference to get the most kind of statistical power. So our idea is to avoid data splitting completely and to leverage something that I've been working on in my research group for several years now called mini patch ensemble learning. And the idea is to take random tiny subsamples of the observations or rows and the features or columns at the same time. So if you permute the data matrix, it's like this little patch right here. So we call this a mini patch. And you're going to do this many, many times. Take these many random tiny mini patches. So it's kind of like double bagging. Bagging is like bootstrap resampling or bootstrap aggregation, but we're not exactly doing bootstrapping, we're doing subsampling, but we're doing it on both observations and features simultaneously. Okay, so this is kind of, again, you can think of this as just bootstrap aggregation, but a little bit on steroids and with very tiny subsamples of observations and features. So we call this mini patches from mini batches, which are tiny subsamples of observations from stochastic optimization, and patches from image processing. And the inspiration it comes from random forest, stochastic optimization, dropout, and stability selection. This idea of kind of randomly dropping out some observations and features has been around and comes up in several different models. And there's a lot of computational advantages here. This is obviously going to be, especially if our tiny mini patches or if our mini patches are very tiny, it's going to be fast to fit any model to these tiny mini patches. It's completely distributed, so it's super easy to do this in parallel, and it's very memory efficient. If you actually have a ginormous data set that you can't even load into memory, you can easily actually load each mini patch into memory and do something on the mini patches and then aggregate the results. Okay, so there's a lot of advantages of that. So the question is if you look at these mini patches, these are random double subsampling. And look, we're leaving out a bunch of observations here and a whole bunch of features here are left out. So the question is hey, this looks like we're leaving one covariate out. We're certainly leaving several features out. And also we've got left out observation sets and the question is, can we leverage these to use as calibration sets for inference? Okay, and so here's what we're going to do. Step one is to fit a mini patch ensemble. And you can use any base model. So the base model could be decision trees, and then you get kind of a mini patch random forest model. You could do neural networks or kind of deep neural networks on each mini patch, and then you get kind of a deep ensemble model. So many different ensembles here. And then we're going to do an inference approach. And here's how we're going to proceed with inference. First, we're going to calculate the lieu predictor. So this is the leave one observation out. And what we're going to do is we're going to sum over all mini patches that don't contain observation I, where observation I was not included in training. Okay, so we can get this kind of Lu estimator, and then we can get the local Lu estimator. And the idea of this is we're going to sum over all mini patches that did not contain observation I or feature J, where these were both left out. Okay? And based on these, we're going to propose a feature occlusion score that's just the difference in prediction errors between this local Lu predictor and the Lu predictor. Okay, and then we're going to construct asymptotically normal confidence intervals for delta J. That's all we're going to do here. So notice all the computational stuff happens in step one. And step one, the mini patches kind of take care of all of the refitting that has to go on here, right? Like, so all of the fact that we're refitting for lots of different datas, these are actually done on very tiny subsets that are really fast. So it's kind of very fast computationally. And after step one, this is just actually summing over indices, right? So it's essentially free computationally to actually do this inference approach, okay? And then based upon our mini patch feature occlusion scores, we're just doing a standard normal confidence interval here for delta J. Okay, so this is just our standard normal lower and upper confidence interval, two-sided confidence interval. Okay, I'm going to pause here. Any questions on this procedure? Okay, so of course you might say, well, this sounds like a cool computational trick, but if you think about this a little carefully, you might be like, well, there's no way that these could actually provide valid coverage of your confidence intervals. And there's two main challenges here. First of all, the delta hat J's are completely very, very dependent because each pair of mini patches depends on the same n-2 samples. And you can show like all the existing central limit theorems really like cannot handle this form of extreme dependency. Like we looked actually when we first started doing the theory for this project, my postdoc at the time, Lily Zhang, was looking at this theory, and she came back and a week later and she said it's absolutely impossible. And then she came back a month later and she said, no, it's completely impossible. You can never do this. And it turns out we have a trick to avoid this issue. But the second issue is also a major challenge because if you think about it, the exact same data was used for training and for inference here. We're actually making full use of the entire data set for both training and inference. And we show in the paper this actually leads to a very new type of selective inference problem that we're proposing here. So to prove the validity of our mini patch confidence intervals, we had to address these challenges. And under these four assumptions, we can actually prove validity. So assumption one is on the prediction error function. It needs to satisfy a Lipschitz condition. This is very trivially satisfied. So this is a non-issue for most kind of error functions you would want to use. We need to assume bounded mini patch predictions. That is, any two mini patches can't have predictions that are infinite. So B just needs to be bounded. And some light assumptions on the size in the mini patches and the number of mini patches required. And under these assumptions, we have asymptotic normality of our delta J bars. And of course, we have that our confidence intervals have asymptotically valid coverage for our feature importance scores. So you might say, how do we do this? And so just very lightly, the proof sketch is actually, there's a very interesting recent paper by Bale et al. That shows shows a central limit theorem for cross-validation. However, they rely on algorithm stability, which is a rather strong assumption to show this. And the trick we used here is we showed that mini patch ensembles are actually stable with any base model, including neural networks, decision trees, whatever you use. The fact that we ensembled them together in diverse ensembles reduces the variance and automatically satisfies algorithm stability that allows us to get around these issues. Okay, and then we can characterize specifically the conditions under which that happens. So that's a little bit of the proof sketch there. So our paper, if you're interested in technical details, I am barely even scratching the surface of the technical details. We actually talk about solving the vanishing variance under the null, which is a problem known in this area of inference. We actually have alternate assumptions that allow you to use any mini patch size you want. We actually prove valid coverage after hyperparameter tuning, so you can tune the mini patch size if you choose, or you could tune your machine learning model within each mini patch and still have valid coverage. We actually analyze the conditions under which it's similar to conditional independence testing and relate this to the post-selection inference literature as well. So just a preview of the simulation studies. Here I'm showing coverage here on the y-axis. So our target was 90% coverage. And on the x-axis, I'm increasing n from 250 to 2000. And loco mini patch is in red, and we're comparing to loco split, and loco split fit on mini patch ensembles. And we see that loco mini patch has almost over coverage in red in all these scenarios. So this was random forest and ridge under several different models, underlying models. We also show the interval width, and here's where you really see the huge increase in statistical power from avoiding data splitting. So here again is the log interval width of our confidence intervals and n on the x-axis, and we see that our loco mini patch in red has significantly smaller confidence intervals, which translates to a huge increase in statistical power compared to loco split and loco split mini patch in pink and in purple here. And you can really see this for a random forest. So our loco mini patch confidence intervals here are in red, and the loco split intervals are in gray here for the same features that have signal strength 3 to 1 and a bunch of null features. And we really see here how much smaller our confidence intervals are, meaning a huge increase in statistical power compared to loco split. So loco split in this example only finds, the way to interpret this, sorry, the 0 line is here. So if these confidence intervals are above 0, we would say that that's a statistically significant feature importance. So for example, loco split here finds that the first feature is statistically significant, but misses all of these others, whereas our loco mini patch has a much more statistical power and smaller confidence intervals and finds other important features. Okay. So I also just want to give you a preview of how we're applying this in science. So here we're applying this to genomics of cognitive decline. We built a random forest model where we're trying to predict which genes are actually predictive of declining cognition in older adults based upon bulk RNA sequencing on postmortem gene expression. So this is a very, very hard predictive task. And here we actually find 3 genes that are statistically significant with loco mini patch. I should say, loco split found 0 genes statistically significant, didn't have enough power. And to understand whether we actually found significant genes, we fit kind of a random forest model with genes 1 and 2, 1, 2, and 3, and so forth to the number of features to 50. And we're comparing against other, these are other different population feature importance methods applicable to random forest here. And we see that our test mean squared error is significantly lower right at 3 genes, which is what we predict to be statistically significant. And this is indicative that we found the right 3 genes using our method, and these other methods really did not seem to find the correct genes. Again, a huge difference between loco split found 0 significant genes, and we found 3 significant genes here. And the first 2 genes, the top 2 genes we found, have already been implicated as related to Alzheimer's disease. The third gene we found is potentially very interesting, is a long non-coding RNA And specifically, this has been previously linked very recently to other neurodegenerative diseases like Parkinson's disease, but there's currently no known links to Alzheimer's, but so this is a potentially very interesting new target to study. So that's kind of our inference approach for feature importance, but I do want to, with the remaining time, jump to kind of higher order feature patterns and feature interactions. And the goal is, can we find these feature interactions, and also can we quantify their uncertainty? So you might say, well, why feature interactions? So these are really important in omics, so understanding how genes interact, proteins, chemometrics, and material science, understanding how two materials interact to create kind of certain properties of materials, understanding how certain drugs and certain molecules are interacting to get an effect for drug discovery and advertising, clinical diagnostics, and so forth. So there has been some existing approaches for this. In the interpretable machine learning community, basically everybody argues that feature interactions are super important and everybody wants to interpret feature interactions, but they say that computationally, these are very, very hard. And so there's been two methods that have been proposed that are model agnostic: the H statistic by Jerry Friedman and others at Stanford, and the Shapley interaction scores. And there's been several versions of these Shapley interaction scores. However, these are computationally incredibly burdensome. And by incredibly burdensome, I mean, when we ran these for p choose 2 interactions, when p was equal to 20, so 20 choose 2 interactions, these took over a day to actually run for those. So meaning it's completely impossible to run these on a reasonable size data set. Now, there are methods specifically for random forest that search for interactions within random forest, and none of these methods permit any statistical inference or uncertainty quantification. So we're going to try to leverage our loco method and our loco inference to actually extend this to interactions. And so the idea is, well, we previously left one covariate out, so why not leave two covariates out? And specifically, we want to evaluate the difference in prediction error when we leave out feature J and K at the same time versus the error of the full original model. So if you stare at this delta JK long enough, you'll see that this is positive when leaving features J and K out increases the prediction error, meaning features J and K are likely important. Okay, now just because this delta JK says features J and K are important doesn't mean they interact, however, because it could be that J and K are both individually important. So to actually define our interaction loco score, we define it as follows. The delta J, so this is leaving out just feature J, leaving out just feature K, minus leaving them out both. And you can kind of think of this as an ANOVA with interactions with a sign difference because we're dealing with prediction error. So we want the error to be low. And if you stare at this long enough, this kind of iLoco metric here, this is the error of this term minus the error of the full model. And this term here is exactly that kind of ANOVA model, the kind of individual effect of feature J, individual effect of feature K, minus the effect of both J and K. Okay, and we show that if you assume a functional ANOVA decomposition, which is a standard kind of assumption in interpretable machine learning for features, iLoco JK does isolate the contribution of the JKth pairwise interaction term. And so positive values of iLoco JK indicate an important interaction, but we also prove another property. We show that negative values of this metric indicate that there are two important features that are correlated, but that don't directly interact. So this is another thing. I'm not going to show results on the second property today, but you can see this in the paper. Okay. We can also extend this to higher order settings other than pairwise, so tertiary and so forth. I'm going to skip this for now. And I'm going to skip our exact kind of estimation and inference procedure, but basically we're going to leverage our mini patches again also to do very fast computation because we can leave out multiple features, two features at the same time, And we can extend our previous inference results to this setting as well. So just to show you, this is showing you the success probability of detecting the top pairwise feature interaction versus the signal to noise ratio of kernel ridge using RBF kernels, an MLP multi-layer perceptron deep neural network, or a random forest model. And our methods are in blue and red, and the other methods are these other colors. And you see that blue and red, as the SNR increases, really dominates all these other methods. So just the metric itself is actually performing better than everybody else. And in terms of computational time, the H-score and the SHAP score took over 24 hours to run for this simulation versus 20 seconds for our model. Okay, so that's a huge difference in computation time. So ours is actually legitimately very fast to compute. We also show valid coverage, but I'm going to skip this for now. Because I do want to end with a kind of fun example of feature interactions and how to use this type of inference. So my husband is very into mixology and making cocktails, and I know nothing about this. However, I'm a willing guinea pig to try any of his fantastic concoctions. And so he's always trying to find cool and new, interesting new ingredients or new ways of combining them to yield the best cocktails. So the question, he actually posed this to me a while ago, and I realized that, wait a second, I can use my method to address this. And his question is, which cocktail ingredients or combinations of cocktail ingredients yield the best drinks? Okay. So to do this, we actually look at Difford's Guide for the Discerning Drinker. And if you're familiar with cocktail making and mixology, you might be familiar with this website. My husband tells me this is a very thorough and detailed website about making cocktails. And so he actually helped me do this. So because he web scraped Difford's Guide's website, the entire website, to pull down almost 16,000 cocktail recipes. And we studied the top 100 cocktail ingredients. And the goal was to use the ingredients to predict Difford's Guide rating. So it's a rating between 1 and 5 stars. And so we fit a mini patch ensemble of deep neural networks. So this is a deep ensemble method. And so that was kind of our model for doing this. So when we fit these results, so first here's our inference for feature importance. So this is loco mini patches, leave one covariate out mini patches. And what we're showing here is the loco score. So the relative scale of the y-axis is not important because it really depends on the scaling of the data set and the error metric. So ignore the scale here, but just look at the relative differences here. And in the x-axis, we see our different cocktail ingredients. And we see there's 4 that look really statistically significant here. And those 4, if we examine them, are lime juice, lemon juice, sugar syrup, and gin. 4 pretty good, I would say, cocktail ingredients. That sounds like you add a little bit of tonic water, and there we have a great G&T, right? Pretty good cocktail ingredients there. But of course, this doesn't look at, these are just individual cocktail ingredients. And the question is really like, what's the coolest interactions? And like, what are the combinations of cocktail ingredients that make for really great drinks? And so here, we're actually showing the interaction loco scores on the y-axis versus combinations of feature interactions on the x-axis. And here we're ordering them by the iLoco metric and showing the confidence intervals here. And notice the interaction coffee liqueur and espresso coffee that has the highest iLoco metric has a super wide confidence interval that crosses 0. So this interaction is not statistically significant. And to me, this really motivates the need to do uncertainty quantification. If we just reported the iLoco metrics, you would think coffee liqueur and espresso coffee are the most important. But clearly there's a lot of uncertainty, meaning there's a lot of variability in this metric and how it affects the ratings of the cocktails, and hence really wide confidence intervals. And another really wide confidence interval here is cream and creme de cacao, sorry. So some of these you can understand have really wide confidence intervals, which just highlights the need for doing this type of uncertainty quantification. But we can examine, so there were several statistically significant interactions, and we can actually examine these. And the top one is absinthe and chartreuse green, which are two maybe rather esoteric combinations that you wouldn't really think of at all, but it turns out to be the most statistically significant interaction in this data set. And it turns out there's a lot of fantastic cocktails from Difford's Guide based on these. These all look like fantastic drinks that use absinthe and chartreuse, and several different possible combinations there. And so my husband made me one of these, and I really liked Neptune's Wrath, which is if you need kind of one recommended cocktail from our exploration of cocktail ingredients, this might be a very fun one to try. And you can even set the absinthe on fire and make a flaming drink out of this if you want some extra drama. Okay, and there's several others that are interesting. So orgeat syrup and gentian liquor. This is maybe a little esoteric. This is almond syrup, and this is a French liqueur. However, lemon juice and tequila here, these are, of course, margaritas. So the third kind of most significant combination really comes up all the time in margaritas and makes for great margaritas. So you see these in a lot of margaritas and margarita knockoff drinks. So hopefully this kind of illustrated, this was just a fun example to show why we need kind of these types of uncertainty quantification for feature interactions and for feature importance and feature interactions, and allowed us to find some kind of fun cocktail combinations and new cocktails that you might not have necessarily thought of as your standard cocktails. Okay, so I'll quickly summarize here. So hopefully I've really motivated the need to do uncertainty quantification for machine learning interpretations, and specifically for interpreting features, feature importance and feature interactions. And hopefully I motivated that there's a lot of variability in these interpretations, and if we just report the feature importance metrics, we might be getting the answer wrong because those might not be the most statistically significant given the variability that we see in these metrics. I also developed a mini-patch loco inference method that is model agnostic and distribution free, completely avoids data splitting, which I think is a huge bonus, and it directly does inference on the learned machine learning model. And there's a lot of theoretical contributions in this paper that might be of independent interest in the algorithmic stability and post-selection inference and also machine learning inference literatures. We also proposed an iLoco metric and inference via mini patches. This is very fast and powerful and performs a lot better than existing approaches. It's computationally feasible to apply for even really big data sets like that cocktail data set. And so that's a really fun new metric there. And I would say we've just barely scratched the surface. There's absolutely tons of future potential for statistical work on inference for other forms of machine learning interpretations. So I think this is really just the start, and there's tons of open problems there that I'd be happy to discuss. So I want to acknowledge my three students or former students and postdocs. So Lily Zhang was a former postdoc. She's now at UIUC. Luchen Gan got her PhD at Rice, and Camille Little is a current PhD candidate at Rice. And Michael Wayland, my husband actually web scraped the cocktail data set, and Tarek Zikri worked on the reliability study as well. And here are some key references and papers on these. And I thank you very much for your attention, and I'm super happy to take questions.
Thanks a lot, Genevera, for this wonderful talk. Are there any questions in the audience? If so, please feel free to write them in the Q&A or raise your hand and unmute yourself. If everyone's shy, I'll start. I was wondering, so when you had this loco for where they were doing the data splitting, which seemed to have much wider confidence interval. Do you think that's because of is it just due to the fact that you're splitting the data in half? And your method is much shorter intervals because of the repeated measure? Because it seems like they have, you have better, higher coverage,
Yeah.
but shorter interval.
Yes.
Is
Yeah,
there any?
and yeah, so what's happening there is kind of a twofold thing. So first, you're only using half your data to train your machine learning method. So your F hat is probably more variable and is not as good, right? And so that is actually contributing to some of it, whereas we're using the full data to train F hat. And second, they're only using half the data for inference. And of course, we know our confidence intervals scale with the size of square root of n. And here they're scaling with square root of n over 2 for data splitting versus square root of n for ours, which does make a really big difference, right? So that's why you see this very big discrepancy in statistical power.
And the fact that your ensemble is more centered around the truth than the
Yeah.
data
Well,
splitting is because of this averaging property as well. I
yeah,
guess.
we're essentially, by doing ensemble learning, we're dramatically decreasing the variance, kind of law of large numbers. We're getting at that target F hat quite a bit better than just fitting a single model.
Thank you. Very interesting. Any other questions?
Oh, hi, thank you for the talk. I was just wondering, for your investigation in ROSMAP, you said you identified 3 genes that were significantly related to cognitive decline. I was wondering, out of how many, if you investigated all the like 15,000 protein coding. I think you also had non-protein coding genes, and also which models you used for that analysis.
Yeah, no, that's a great question. So I've analyzed the ROSMAP data in several other papers. For this study, we actually significantly did some feature filtering. So we didn't fit all, I think it's like close to 50,000 transcripts in that data set. And we did not, we did a lot of feature filtering, less for our method, but for the other methods that we did for comparison, because they're computationally very intensive. So I think we filtered down, if I remember correctly, to only the top like 100 genes based on variance, so high variance kind of transcripts. So again, this I think wasn't a particularly rigorous study, but even we found 3 interesting genes for a very hard predictive problem, even with just doing a simple variance filter. I'd love to go back with just our method and analyze the full data set.
Would that work for like thousands of genes?
Yes, you can absolutely. We've applied our data set to on the order of, tens of thousands of features and hundreds of thousands of observations, and it's computationally, again, very fast. As long as you can distribute things, as long as you have access to a distributed system, it's computationally very fast to run.
Okay, thank you.
Yeah, someone is asking to show the slide with the papers and the reference.
Oh, the paper.
Another
Okay,
question to know if it's possible to share the slides afterwards. I think people were interested in
Okay.
references there.
Thank you so much. Thank you so much for this really interesting talk. I have so many questions. It was really informative and I really liked it. And I'm gonna start with like an easy one, which was on, I found your study of stability of the importance measures to be really interesting. And I was curious if you were focusing like on the values of the importance measure, or if you were focusing rather on the ranking of the features. And if you only focus on the ranking, is there such a discrepancy?
Yeah, so the results that I presented were for the ranking of the feature importances, not the feature importance values itself. And we did that for a reason, and that's because we feel like that's the one that's the most comparable across methods. Like we couldn't figure out a way to actually compare the feature importance metrics, which are on wildly different scales across methods. So, but the feature ranking is comparable across methods. So we actually looked at the rank of the feature importance scores using the rank biased overlap, which weights the top ones a little bit more as it goes down, kind of the ranking. So that's the results that I presented that showed that the feature importance metrics are very unstable.
Okay, great. If there's no other question, I can have a second one, if that's okay. Yeah. Great. So yes, I was wondering. I wanted to pick your brain on a kind of a question that we stumbled upon during our research, which was trying to disentangle the effect when we're dealing with interactions, disentangle the effect of dependence between inputs and like the functional effect of the model itself. And I wanted to know if you had something to say about it, if you have.
Yeah, so a little bit, I'll go back if I can find the slide here. And yeah, so specifically, we found that like correlation does affect kind of our interpretation of interactions also. And specifically, like this metric has a very large positive value when two features interact that are not correlated. If they are correlated, it will still have a positive value, but it won't be as strongly positive. And actually, this has a very interesting property that we show, which is a lot of feature importance metrics are known to behave terribly with correlated features. It's a big problem that's well known in the literature. Here's a reference if you want to read about how terrible everything is. And what's really cool is this, if we have two important features who have like 0.99 correlation and they're both important, if you just do loco itself, loco will they will compensate for each other and the loco scores will be very, very low. However, the iLoco scores will be extremely negative. And so we can actually use these to pick out these negative scores to figure out the important correlated features. And so we do want to explore this property further because I do think this is a really interesting facet of these types of like iLoco scores.
Okay, thank you. Thank you very much for the answers.
Okay, so if there are no more questions, thank you everyone for attending this great talk, and join me in thanking our speaker. And I'd just like to let you know the next colloquium in statistics, machine learning, and so on will be by David Landrio on March 21st, and it will be live at Concordia University. So stay tuned, and thanks a lot again, Genevera, for the talk.